नमस्कार, मैं आनंद कुमार खत्री, एसोसिएट प्रोफेसर ऑफ जूलॉजी, गवर्नमेंट डूंगर कॉलेज बीकानेर। आज के हमारे लेक्चर का विषय है फर्टिलाइजेशन, पार्ट सेकेंड, एक्टिवेशन ऑफ ओवम। द फिनोमेना ऑफ एग एक्टिवेशन involve changes in ionic permeability of egg plasma membrane due to activation of various pumps such as proton pump, sodium, potassium pump, etc. Transient increase in intracellular calcium and pH, the cortical reaction and elevation of fertilization membrane, fertilization, cone formation, cytoplasmic rearrangement, changes in physical properties and metabolism of the egg, resumption of meiosis and preparation for mitotic cell division for cleavage. This is the diagram which showing the stage of sperm egg association during fertilization of polychaete annelida hydroids, showing many acrosomal tubules. First, changes in ionic permeability and potential of egg's plasma membrane. The change that occurs at the point of membrane fusion is transmitted as a wave or impulse over the egg surface and subjacent cortex. The egg surface undergoes a change in ion permeability accompanied by altered electrical potential properties of the membrane. Electrical potential changes at the moment of fertilization resemble changes occurring in a nerve when an impulse passes over it. In the egg, however, the time constant is much longer than it is in a neuron. In the starfish astrias, for example, the inside of the egg has a membrane potential of -30 to -60 millivolt, and at fertilization the potential decreases about 5 millivolt, a change lasting about 1 minute. Larger drops of membrane potential are seen in eggs of the fish and toad, and magnitude of the potential change varies with the ion contents of medium. If more sodium is added, the potential drop is reduced, while the addition of potassium increase is the change by 10 to 15 millivolt. The response of these ions suggests that the primary activation event of the egg is a change in ion permeability at the site of sperm contact. Sodium, potassium and calcium ion diffuses rapidly into the frog egg, reaching a peak within a minute after sperm attachment. The permeability changes in then propagate as a wave over the membrane within a matter of seconds and involve a variety of transport proteins or pumps of membrane. This is the diagram, acrosome reaction and sperm's entry in ovum of rat. B. Transient intracellular rise in calcium ion. The first directly detectable intracellular changes is a transient rise in calcium ion released in the egg cortex from intracellular calcium store, which can be recharged after 40 minutes. The rise begins 20-25 seconds after sperm fusion and lasts for 2-3 minutes. When the basal level are again attained, the transitory Ca++ increases insufficient to cause the cortical reaction not only in sea urchin but also in fish eggs. The role of calcium ions in fertilization. Of all the ions involved, calcium seems to play a major role in egg activation. In the absence of calcium, fertilization does not occur. Sperm's viability, motility, and fertilizing capacity are also diminished in calcium-free media. Furthermore, the acrosome reaction is calcium dependent. Without it, no acrosome filament explodes. Egg stability also depends upon calcium. In its absence, changes in viscosity and rigidity of the cortex are observed. This is the diagram showing the change in membrane potential of the starfish Astria's egg at fertilization. Changes in the distribution of calcium within the egg are noted at fertilization. Calcium seems to be found to the plasma membrane and cortex, but after membrane fusion, calcium is released, becoming freely diffusible within the cytoplasm, moving from one compartment to another. This calcium activation event may change protein configuration in membrane and cortex, with consequent changes in functional properties. Thus, calcium ion may be involved in maintaining protein configuration. Any changes in its association with proteins changes protein function. Conformational changes of membrane proteins, that is, account for increased permeability for sodium, potassium ion, and other substances. Finally, calcium exchange across membrane in excitatory event is coupled to cyclic AMP, a regulatory molecule that activates many coenzymes including protein kinase. Protein kinases are enzymes that phosphorylate proteins such as histones and may also phosphorylate microtubule protein that function in cell division. Since histones have a structural and possibly a regulatory role in cell division in chromosome function, we can say that activation of histone and microtubular proteins among other might be essential to further development of the egg. C. Transient intracellular increase in pH. The second important transient change beginning at the same time, that is increase in calcium ions, but lasting for about 10 minutes in the intracellular increase in at pH by 0.3 to 0.4 units. The increase is due to an inflow of sodium and outflow of H plus ion. The latter causes the formation of fertilization acid in the surrounding seawater. Thus, the pH of the cytoplasm increases from 6.8 to 7.27 in sea urchin X. Fourth, D. The cortical reaction. Egg membrane activation is intimately tied to changes in the egg cortex. Within seconds after membrane contact, changes occurs in the egg cortex. The final microvilli of the egg membrane are withdrawn and the egg surface becomes smooth. The egg decreases in volume, that is, it slightly shrinks. The dense granular layer of the cortex becomes a homogeneous region or oriented microfilaments adjacent to the plasma membrane. These changes signal altered physiological state of the cortex. The cortical reaction may differ from one group of animal to the other, but in most groups it fundamentally culminates into the formation of a membrane called fertilization membrane outside the egg plasma membrane. The fertilization membrane blocks the entry of late arriving spermatozoa. The process of the cortical reaction and fertilization membrane formation have been studied more extensively only in certain echinoderms, sea urchin, and starfish. First, fertilization membrane formation in sea urchin. In sea urchin, as soon as the apical end of the acrosomal tubule touches the surface of egg from the site of the contact, a wave-like color changes from yellow to white travels rapidly around the egg cortex and is shortly followed by the elevation of fertilization cone from the egg surface and the formation of fertilization membrane around the egg plasma membrane. Electron micrograph of sea urchins for unfertilized eggs show that the egg cortex is bounded by the two membranes, an outer 30 angstrom thick vitelline membrane and inner 60 angstrom thick plasma membrane. Beneath the plasma membrane occurs a layer of cortical granules, about 15,000 cortical granules. A fertilization membrane is formed in following stages. First, the outer vitelline membrane separated from the plasma membrane undergoes expansion and becomes the outer layer of the fertilization membrane. Second, the cortical granules explode and release following three components: dark, dense, lamellar and fold part of the granules unfolded and fuse with the inner side of the already elevated vitelline membrane. Globules which fuse together and build up a new surface of viscous hyaline layer just at the outer side of the egg plasma membrane. The hyaline layer adhere closely to the surface of the egg, and during cleavage it helps to keep the blastomeres together. The liquefied components of the cortical granules fills the previtelline space. It contains mucopolysaccharides and abundant water. The contents of cortical granules also includes a trypsin-like protease enzyme. All these structures, namely vitelline membrane and contents of cortical granules, forms a fertilization membrane. Ultimately, the membranes of cortical granules fuses with egg plasma membrane to form a mosaic, such as an altered membrane may contribute to the change in permeability. Grant has speculated that cortical granules membrane of an egg are already differentiated as special transport systems, so when they are fused with the plasma membrane they are activated to alter the permeability of membrane. The sequence of cortical granule fusions with the egg membrane resembles an enzymatic activation traveling as a wave over the egg surface from the point of sperm's attachment. It is in sea urchin's egg propagated over the surface in 20 to 30 seconds. One hypothesis for impulse spreads is that a self-propagating chain reaction is initiated by enzymes formed at the site of the sperm's attachment, which diffuse over the egg surface and initiates cortical granules breakdown. For example, minutes after fertilization, lipases and proteases become active and they may digest cortical granules membrane to initiate breakdown. Another enzyme activated at fertilization is a calcium dependent adenosine triphosphatase, which could explain the requirement for calcium at fertilization. Splitting the ATP may furnish energy for explosive release of granules or for contraction of the egg surface. Among other invertebrates, cortical granules reaction and fertilization membrane formation have been observed only in certain animals such as nereid and annelid worm. In fact, there are conflicting opinions as to the significance of the cortical granules reactions at fertilization. In some eggs, such as the barnacle Barnea, cortical granules explodes independently of fertilization. In the annelid worm Chitopterus, cortical granules do not break down at fertilization but are retained during early cleavage. Second, fertilization membrane in vertebrates. Comparable changes occurs in the cortex of different species similar to sea urchin. In bony fishes and frogs, the cortical granules contain mucopolysaccharides. In the eggs of these animals, the cortical granules are broken after sperm's penetration into the egg cytoplasm. Their contents become liquefied and extruded on the surface of the plasma membrane of the egg. They gradually fill up the perivitelline space which is already occurring in between the chorion and egg plasma membrane in case of bonifaces, or the space lies in between the vitelline membrane and the egg plasma membrane in case of frogs. In both cases, the vitelline membrane or chorion itself does not transform into the fertilization membrane like the sea urchin. The unfertilized egg of the some mammals, man, rabbit, hamster, have cortical granules in their cortical regions. In them, the sperm penetration is not followed by the formation of fertilization membrane, but the cortical granules burst open and release their contents into the perivitelline space, that is the space between the egg plasma membrane and zona pellucida. They ultimately disappear. Lastly, there are many vertebrates such as chordate or urodales, amphibian, mammals, etc., which lack cortical granules. In these animals, no fertilization membrane formation occur. However, in chordate amphibians, a hyaline perivitelline space is formed in fertilized egg. This is the diagram showing the change of the egg cortex of sea urchin. Next, monospermic and polyspermic fertilization. In most species, successful fertilization occurs when one and only one sperm enters the egg. This is called monospermic fertilization. When more than one sperm enters, a genetic imbalance is created and development is abnormal. In 1902, Boveri noted that dyspermic sea urchin eggs develop into bizarre forms. He could show that the first division of the egg was abnormal because a multipolar mitotic spindle forms by two sets of centrioles donated by two sperms. The triploid chromosomes complement was randomly distributed to 3 cells at first division, and subsequent cells were aneuploid and developed abnormally. When any spermatozoa penetrate the single ovum, this is called polyspermic. When many spermatozoa penetrates the single ovum, this is called polyspermic fertilization. It is of two types: pathologically polyspermy, bispermy or trispermy occurs in some mammals and sea urchins if the immature or aging or overripe egg is put in a dense sperm suspension. This is called pathological polyspermy. Second, physiological polyspermy. In some animals, particularly those with much yolk in their eggs, that is, in polylacethal eggs, in some insects, elasmobranchs, urodels, reptiles, and birds, and also in microlacethal eggs of bryozoans, the egg is naturally entered by several sperms. This is called physiologically polyspermy. In these cases, genetical material from only one sperm is incorporated in the zygote nucleus and other sperm's nuclei are degenerated. Mechanism to block polyspermy in monospermic forms. The mechanism involved in checking of polyspermy is best studied in some sea urchins. There are two main mechanisms to prevent polyspermy. A. Fast block to polyspermy operative from 3 to 45 seconds after the addition of sperms is due to early inflow of ions and membrane depolymerization. A slow block to polyspermy starts about 30 seconds after sperm's addition and lasts till hatching. It is due to A. A steric effect because the cortical reactions elevates a fertilization membrane which physically prevents the sperms to reach the egg surface, and B. An enzymatic effect of trypsin-like protease is released from the cortical granules which degrades binding receptors from the vitelline membrane and detaches the bound sperms. All these mechanisms together are indeed very effective blocks of polyspermy. In other words, in other species, the block of polyspermy is only partially understood and is also either to membrane depolarization fish or elevation of fertilization membrane or both in rabbit. It in some fishes and in some fishes, the micropyle is plugged. Ascidian eggs rely on the chorion or tough outer membrane to prevent multiple sperm entry. In mammals, the zona pellucida prevents polyspermy. In the latter case, penetration of one sperm changes the organization of zona so that no other sperms can penetrate. Sixth, metabolic activation. In mammals, in which the egg before fertilization become metabolically inactive, the fertilization leads to a sprout of metabolic activity. Metabolic activation of the fertilized egg is depicted in the following changes. First, A. Respiratory change. In a fertilized egg, the rate of respiration either changes or decreases or remains static. This appears to be a relation between the post fertilization oxygen consumption and the stage of maturation of the egg at fertilization, because at the time of the fertilization the sea urchin has completed maturation. The eggs of buffo is at second maturation division stage, and the egg of Cytoperus is at the first maturation division stage. The increased oxygen consumption is related with the oxidation of glycogen and other foodstuffs of the eggs and synthesis of numerous ATP molecules. B. Coenzyme changes. In a fertilized egg occurs a interconversion of pyridine nucleotide enzymes NAD into another coenzyme NADP and NADPH due to phosphorylation of the NAD in the presence of an enzyme NAD kinase. There are ample evidence that NAD kinase enzyme, though present in the unfertilized egg, it exists in an inactivated state. It is made activated only at the time of fertilization. Appel and Inverson 1965. Since the coenzymes NAD and NADP are involved in numerous enzyme mediated oxidation and reduction reactions of the cells, and because of their obligatory role in accepting or donating electrons for these reactions, any changes in concentration or availability of these enzymes may have profound metabolic consequences, and the increased NADP and NADPase contents may initiate many synthetic pathways of fertilized egg. Appel et al. 1969. Next, C. Changes in the rate of protein synthesis. The cytoplasm of the mature unfertilized egg, though contains complete machinery of protein synthesis such as DNA molecule, tRNA, mRNA, ribosomes, and enzyme required during protein synthesis, none or virtually very little protein synthesis occur in it. There are evidence that during the later phase of oogenesis, some inhibitor or repressor proteins are manufactured in sea urchin's egg, and these proteins inactivate chromosomal genes, mRNA proteins, ribosome, etc. Metaphora et al. 1971. During fertilization, some proteolytic enzymes remove these inhibited proteins from them and activate protein synthesis. Thus, before fertilization, the ribosomes in such sea urchin egg cytoplasm are single. After fertilization, increasing quantities of ribosomes are joined in groups which show they are actively engaged in protein synthesis. Balansky 1981. After 5 minutes of fertilization, the rate of protein synthesis increases 3 to 12 fold about 20 minutes post fertilization. The DNA synthesis in the pronuclei begins. The protein and DNA synthesis herd cleavage and development. Seventh, resumption of meiosis. In most animals, meiosis is arrested at a particular stage and resumed only after fertilization when the polar body extruded. The time of fertilization varies from species to species, and it has been found that the spermatozoa may enter the egg at different stages of maturation in different animals, as it has been seen, tabulated in following page. This is the table showing the stage of egg at which fertilization occur in various animals. Next, eighth, initiation of mitosis. The most significant aspect of egg activation, that is the initiation of mitosis for cleavage. The initiation of mitosis takes place due to following agencies. A fertilization, the turning on the DNA synthesis take place. Immediately after fertilization, the rate of DNA synthesis increases with greatest pace. It has been observed that in most of the animal eggs, the fertilization is followed by the selective accumulation of uptake of cytoplasmic DNA polymerase enzyme, an enzyme used in DNA synthesis by the nucleus. Recent investigations show that a spermatozoa must introduce its own centriole if the egg is to form a mitotic spindle and divide in normal way. The unfertilized egg cytoplasm though has centriole, but the centriole is incapable to divide and also to form a mitotic spindle of microtubule. Next, ninth number, theories concerning the activation of egg by sperm. It is still not very clear that in what manner sperm activates the egg during fertilization. The following theories have been given or forwarded by different embryologists to explain the mode of activation of egg. Here we just only taken the name. Biomolecular theory of activation given by R.S. Lilly 1941. Second, sensitization of calcium theory given by Brackett 1936, Alban 1930-40, and next, third, Loeb theory given by Loeb 1964, repressor theory of activation given by Monroy 1965. This is the amphimixes in sea urchins and the track of pronuclei of the sea urchin and their movement towards each other and to the site of the cleavage nucleus, with sperm entering or at opposite side of the egg from site of egg nucleus and form same side respectively. Recently, studies using video microscopic data and microelectrode to record bioelectrical responses have shed some light on the initial phenomena that activates the egg. It appears that after acrosome reactions there is an elevation of calcium ion and an increase in pH inside the sperm, and that localized increases in the cytoplasmic Ca++ at the place of sperm and egg contact is sufficient to trigger the exocytosis of the cortical granules and elevation of the fertilization membrane. This is followed by a propagating wave of Ca++ released from intercellular stores and by an increase in the cytoplasmic pH. These two components, Ca++ and pH, appear to be the regulators that trigger the various cytoskeletal changes occurring in fertilization. In addition to protons and Ca++, columbidian proteins and possibly cyclic nucleotides are also involved in these complex molecular mechanism. Migration of pronuclei and amphimixes. At the times of penetration of spermatozoa inside the egg cytoplasm, the sperm's nucleus remain compact and its mitochondria and centriole remain situated behind it. To perform the act of amphimixes, the sperm nucleus has to perform two activities. First, it has to become pronuclei, and it has to migrate from the site of fertilization to the site of amphimixes. As the sperm's nucleus moves towards inward from the site of the fertilization cone, it soon rotates through the angle of 180 degrees so that its mitochondria and centriole assume the leading position. Beside this rotation, the sperm's nucleus starts swelling and its chromatin, which is very closely packed, becomes finally granular. It ultimately becomes vesicular and has an appearance like the interphase nucleus and is called male pronucleus. At the same time, the sperm's estrus forms in the egg cytoplasm around the proximal centriole of the sperms. As the male pronucleus develops and migrates towards the site of amphimixes, the sperm's estrus seems to lead it. The sites of amphimixes lies either near the center of the microlecital and mesolecital eggs or in the center of the active cytoplasm at the animal pole of macrolecital and telolecital eggs. As the sperm's pronucleus and centriole moves inward, it may be accompanied by some cortical and subcortical cytoplasm. If the latter is heavily pigmented as an amphibian egg, the trajectory of the sperm pronucleus may be marked by pigmented granules trailing along its path. This is called the penetration path. Before amphimixes, the nucleus of the egg also undergoes certain changes like the sperm's nucleus, and after the completion of second meiotic division, the haploid nucleus of the egg occurs near the surface of the egg in the form of several vesicles known as karyosomes. In a fertilizing egg, these karyosomes fuse together to form a female pronucleus, which swells, increases in volume, and becomes vesicular. It also migrates towards the site of the amphimixes. The fusion of male and female pronuclei is called amphimixes. The actual fusion of the male and female pronuclei may differ in detail in different animals. In most animals, the two pronuclei actually fuses together, that is, the nuclear membrane of both pronuclei are broken at the point of their contact, and the contents of both pronuclei unite into one mass, which is ultimately bounded by a common nuclear envelope. And in other cases, ascaris, some annelids and mollusks, however, the male and female pronuclei do not fuse as such, but the nuclear membrane in both get dissolved and the chromosomes are released. In the meantime, the centrosome of the spermatozoa has divided in two, and an achromatic spindle is formed, and to this spindle the chromosomes derived from the male sperms and the female pronuclei become attached. Only after the completion of the first division of the first fertilized egg, the paternal and maternal chromosomes become enclosed by common nuclear membrane to form the nuclei of two blastomeres. This is diagram showing the ooplasmic segregation after fertilization in the egg of Stella parcia. Ooplasm segregation. One of the principal results of egg activation during fertilization is reorganization of the egg cytoplasm. This, in turn, may change the original polarity of the egg and affects the future embryonic axis. Many cytoplasmic organelles move about the egg after fertilization to take up new position, often within a matter of seconds after sperm entry. This cytoplasmic restructuring is called ooplasmic segregation, and it creates new axes of polarity and symmetry. During ooplasmic segregation, morphogenetically active materials are redistributed in the cytoplasm and come to occupy specific regions. As a result of some morphogenetic substances are segregated into certain cells during the first few cleavage divisions. Sorting out morphogenetic substance in this way may determine the cell fate early in development, just as it does in the germ line. This is the diagram showing the formation of the gray crescent in the amphibian egg. First, A, 1 of the 2 hours after insemination, before the gray crescent has formed, and the B, after formation of gray crescent, diagram showing the gray crescent contraction of pigmented cortex. The significance of fertilization. First, the gamete or the sperms cells, germ cells, constitute the only physical bridge between the successive generations. The gametes possesses haploid of the chromosomes. The fertilization restores the specific parental chromosome configuration. Fertilization second brings together the chromosomes and gene different lines of the ancestry and results in new genetic recombination which occurs ensures improved adaptations to ever changing environment. Third, fertilization activates the egg to start cleavage. Thanks.